gals. We are diving in and discussing the urinary system, the urinary and digestive systems for this unit. And candidly, Ms. Londrich, we're going back and forth on this one. Traditionally, urinary and reproductive systems are grouped together, but also traditionally, the digestive system is covered before the urinary and reproductive systems. And just with the way that the exams were falling and with our commitment to try to have lecture and lab synchronized as much as possible, we decided to split the difference and do urinary and digestive for this upcoming unit. So as we're looking at our urinary system, this is going to be broken up into multiple parts, primarily because we have a Halloween special coming up a week from today. So that made it a little interesting putting together the presentations. Look at our urinary systems. The big idea, the big picture behind the urinary system is really it removes waste products from the body. A lot of students get the function of the liver and the function of the kidneys flop with each other. They have a similar yet distinctly different function when it comes to waste products. The liver removes waste products from the body by chemically altering the waste products so that they're no longer considered toxic, so to speak. The urinary system removes waste products from the body by physically putting it in the urine and it leaves into a toilet. So they both remove waste products, but they remove the waste products very different ways. Now, when we look at our kidneys, they regulate urine production, so they're going to be very intimately related to fluid balances in our bodies. If we change the fluid, we change the volume of blood, we change the volume of fluid, and as we change the amount of blood in our bodies, pressure and overall concentrations, osmolarity, hematocrits, and so forth of the blood will also be altered. At the embryonic development of our urinary and our reproductive system, there's a lot of shared embryonic development, and we'll talk about those in our next unit when we get to the reproductive system. A lot of times they're just combined and called the urogenital system because they're so intimately related with each other. This is a survey for organs of the urinary system. So let's get a frame of reference here. As we look at these structures, let's go with green. The adrenal glands are on top of the kidneys. They are superior to the kidneys. And these adrenal glands are always grouped in with the urinary system, even though they're more of an endocrine gland and an endocrine function. But since they're just right there, we always present them with the urinary system. Then we have two kidneys. Those two kidneys have a tube coming from each called the ureter. The ureter is not the urethra. Every semester I'll typically have, say 15 to 20%. You know, a significant subpopulation of my students will flip-flop ureter and urethra because, hey, they sound really similar, and they both transport urine. The ureter carries urine from the kidney to the bladder, the urinary bladder. The urethra goes from the urinary bladder to the external environment. So that's big stuff, the big parts of the urinary system. Now, when we look at that's where all the action is at. Well, not all, but the vast majority of the action of our urinary system. Our kidneys have a lot of different functions. They do many different things for us. You can take all of those functions of the kidneys and group them all together under the phrase filters and monitors blood. So as we're looking at this process, the blood goes into the kidneys. It's being processed by the kidneys. And our kidneys will make a bunch of executive decisions about our blood composition. They'll do things like remove metabolic wastes from our blood. Sometimes those metabolic wastes are passively dumped into the urine, or I should say filtrate, because it's not quite urine at that point. So they're passively dumped into the filtrate. Sometimes our bodies will actively transport these metabolic waste products into our filtrate. As we change the amount of filtrate and urine, blood volume, blood pressure, and blood osmolarity will all be altered. Just by changing the amount of water in our blood, those three variables will be altered. When we think of this process of selectively filtering our blood, we'll add and remove a bunch of different ions or electrolytes. Sodium and potassium, bicarbonate and chloride are the five big electrolytes. Hydrogen and bicarbonate are primarily associated with the acid-base balance. And then we just have sodium and potassium and chloride as just your regular Joe Schmo electrolytes, or dissolved ionic compounds. We talked about this earlier in the semester on our first unit when we talked about the blood and the cardiovascular system. Our kidneys regulate blood oxygen concentrations. If our kidneys detect hypoxia or in low oxygen blood oxygen concentrations, our kidneys will release a hormone called EPO, called erythropoietin, and that erythropoietin goes to the red bone marrow and stimulates erythrogenesis or erythropoiesis, the production of new red blood cells, to upregulate blood oxygen concentrations. Our kidneys help to regulate blood calcium levels by secreting a hormone known as calcitrol. Calcitrol was one of the hormones of the endocrine system that we talked about, I should say one of the many hormones of the endocrine system we talked about last semester. As we look at our kidneys, they remove excess hormones from the blood. The hormones that are secreted into our body have a given half-life, or they can hang around in our body being metabolically active for a certain amount of time before they start to degrade. The rule of thumb for clearing hormones is that protein-based hormones are cleared pretty quickly because they're soluble in water. And they'll be broken down into amino acids, and then those constituents are then removed from the bloodstream. The one that we have a hard time removing from our body, our kidneys in particular have a hard time removing, are the lipid-based hormones, or the non-polar hormones, the ones that are hydrophobic. But eventually we can clear them from our bodies. We detoxify free radicals. And when we think of free radicals in our bodies, this detoxification process usually involves some sacrificial molecule banging in, physically smashing into the free radical. When we think of a free radical, you could think of it as an unpaired electron in our body. Unpaired electrons, or naked electrons, are really trying hard to find a partner. They're trying to get into that lone electron pair state. Most of the free radicals in our bodies are going to be in the form of hydrogen peroxide, H2O2, or maybe a superoxide. And then finally, when we think of our kidneys, this is kind of a pathway of last resort. They can make glucose. And they make glucose primarily for our brain. Our brain is very glucose dependent. Most of the other tissues of our body, other than nervous tissues, can use proteins or fats or different kinds of carbs to make ATP. But our brain uses so much ATP that it's designed specifically for glucose overwhelmingly. So in starvation situations, our kidneys can literally turn your muscle into sugar, gluconeogenesis, making new sugar molecules. It's kind of like cannibalism, just at a cellular level. So some of the waste products that we need to remove from our blood include the nitrogenous waste products. These nitrogenous waste products primarily come from protein degradation because we have lots of proteins. That over 50% of all the macromolecules in our bodies are protein-based. Some of these nitrogenous waste products do come from nucleotide breakdown. But as we look at these waste products, let's just make sure we can define some terms. When we think of waste, it's something we don't need, something we don't use in our bodies. Metabolic waste is something we make that we need to get rid of. Some of the waste products in our bodies that we get rid of, we don't make. So think of the stuff that you eat that you don't process, it comes out the backside when we think of the digestive tract. Metabolic wastes, though, have a hard time exiting via the digestive tract. Those metabolic wastes are preferentially dumped into the bloodstream, and from the bloodstream, our urinary system, particularly our kidneys, remove the metabolic wastes from our bodies, preferentially. One of the most common nitrogenous waste is urea. And as we look at a generic protein molecule, that generic protein is broken up into its constituents, its amino acids, those monomers that make protein. And then the amino acid will have a carboxylic acid functional group on one side. It'll have an amino functional group on the other side. And that amino group is cleaved and removed. And we have an NH2 floating around. NH2 is then going to be not hydrolyzed, but hydrogenated and have an extra hydrogen added to it to turn it into an ammonia molecule. Having a bunch of excess ammonia in your body is not a good thing. So we need to process that ammonia. Our liver will take the ammonia and reduce the toxicity of ammonia by converting it into urea. And then that urea is then eventually dumped from the body, urinated out. The namesake of urea is urine because that molecule was initially isolated from urine. For those of you who are interested in biochemical history, there are some absolutely disgusting tales about how these first biomolecules were isolated from the human body. Talk to me after class if you're interested. Another nitrogenous product that we need to get from our body is uric acid. And when we think of uric acid, that's primarily going to be from nucleic acid breakdown. When we say catabolism, that's a term from last semester, that's taking a big, complicated molecule and chopping it up into smaller, less complicated molecules. So urea primarily comes from protein. Uric acid primarily comes from nucleotides or nucleic acids. There are some other waste products that we need to remove from our bodies. Creatine being one of them. Creatine, I go back and forth on this one. I remember when I was coaching wrestling and softball, and that was a train wreck. I had never played softball ever in my life, and then I was voluntold to be a softball coach. It was bad. But very specifically, I had all these athletes underneath me, and they were taking creatine supplements, and then they would go lift weights last semester about the physiology behind creatine and how you can get a few more reps into each set in the weight room to hopefully maximize hypertrophy of your muscles. The downside of creatine is that it's fairly toxic to our bodies. So when somebody's taking a creatine supplement, they're encouraged to drink lots of excess water to help filter and get that creatine out of our bodies. Another side effect of creatine supplementing, at least back in the early 2000s with the kind of creatine that was on the market, was that the urine was kind of a phosphorescent green, bright, like safety green urine for students or athletes that took a lot of creatine. That creatine is shown right there for its molecular structure, and it's preferentially removed from the body by the kidneys. We also are going to, as we look at urea and ammonia, the ammonia forms the urea. Ammonia and urea concentrations are directly related to protein catabolism. So if we are looking at blood urea nitrogen, this is going to be primarily the amount of nitrogenous waste in the bloodstream because urea is the most common nitrogenous waste in the bloodstream by far. So yes, there are other nitrogenous wastes. We have a little bit of creatine, we have a little bit of uric acid, but by far the most common nitrogenous waste in our blood is urea. So as a just general rule of thumb, we can just do a urea test. And that's a good way to approximate the overall amount of nitrogen in our blood. For individuals that are in a healthy range, we have, 10 to 20 milligrams per deciliter, so, a small concentration of urea and nitrogen in our bloods. Every once in a while, we have too much nitrogen. That's known as azotemia. This azotemia is related to a potential renal insufficiency. Renal is a term we're going to be using a lot in this unit. It is the technical term for a kidney. You may remember we had the renal artery and the renal vein during week 4 of lab. That's the artery and vein that go to the kidney. For most of the structures associated with the kidney, from a lab sense, you just put the term renal in front of it, and now it's associated with the kidney. We have the renal hilum, the renal columns, the renal pyramids, just kidney stuff. If we have uremia or an inappropriate amount of urea in our bloodstream as well, instead of having an elevated, we have a depressed level, we can have a lot of extra symptoms associated with that, including excessive amounts of diarrhea because our blood osmolarity is altered relative to our digestive lumen osmolarity, cardiac arrhythmias, and other issues associated with the inappropriate amounts of nitrogenous waste in our bodies. The best way to treat this issue is to have a blood transplant, or to have the blood filtered. So there'll be blood transfusions. There'll also be blood filtration processes as well, because most of these issues are associated, not all, but most are associated with some kind of a renal insufficiency, or maybe a high school athlete who was drinking way too much creatine. As we move on, we have excretion. And when we think of excretion, it's a technical term in this context. That means the removal of waste fluids from our bodies. Excretion is the removal of a waste fluid from our body. The fluid that we remove the most of is urine. So when most people talk about excretion, they think urination. But technically, air is a fluid too. We talked about that just with our last unit. So our respiratory system excretes carbon dioxide gas, a waste product from our body. When we think of our skin and the process of sweating from our sudoriferous glands, we excrete waste on the surface of our skin. Those waste products are thankfully recycled and used to make the acid mantle of our skin that makes our skin slightly acidic and highly saline so that most bacteria can't get through that first line of defense. We also are going to have excretion from our digestive system. When people think of the digestive system and the removal of waste, they overwhelmingly think of defecation, because that's the biggest player. The biggest thing that comes out of the digestive system is feces. But a lot of people don't necessarily realize that towards the end of the colon, at the distal portion of the descending colon and in the sigmoid colon and rectum, we have a lot of excretion as well. We need to rehydrate the feces to make sure that it's loose enough to make it through the anus and the anal canal. Something else that a lot of individuals don't necessarily implicitly know, or it's kind of counterintuitive, is that the kidneys are not part of the abdominal pelvic cavity. And I remember I mentioned this last semester. It was like just a brief afterthought when we were talking about body cavities. I didn't spend a lot of time emphasizing it. We're circling around and spending some more time emphasizing this right now. The kidneys are not in the abdominal pelvic cavity. They are retroperitoneal. Another name for the abdominal pelvic cavity is the peritoneal cavity. Our kidneys are posterior to this peritoneal or abdominal pelvic cavity, hence the term retroperitoneal, or behind. So if we look at our abdominal pelvic cavity, I'm outlining the portion of it shown on this figure in green. We have I'm drawing an arrow pointing forward to the anterior side. I'll put a P for the posterior on the bottom of our screen. I'll put an A on the top for anterior. So the abdominal pelvic or peritoneal cavity is up in front in green. And I'll go ahead and circle our kidneys. Let's go with blue. Our kidneys are I thought I said blue. There we go, blue. Our kidneys are posterior to that. So here's a transverse slice through a human torso to show this viewpoint. As we look at the shape and size of a kidney, this might be a chicken named after the bean, or the bean named after the organ. I'm not sure. But they both have the same shape and general coloration to them. So kidney beans look a lot like human kidneys. And I want to emphasize human kidneys because other mammals have distinctly different looking kidneys, if you've done any kind of a comparative dissection course. It's also worth emphasizing that we as human beings are terrestrial organisms. One of the biggest barriers we have as a terrestrial land-based organism is keeping fluids in our bodies. Fish and organisms that live in saltwater or freshwater don't really have well-developed kidneys. They don't struggle with keeping enough liquid in their bodies. So their kidneys are going to be really puny and small. We as terrestrial organisms have rather overdeveloped kidneys compared to our aquatic counterparts. As we look at our kidney, the general size of it, and I really appreciate this analogy, is a bar of bath soap. So you have your brand new unused bar of bath soap. So they aren't actually that large, but they do a very wonderfully significant thing for our body in that they regulate blood fluid volumes. As we look at the lateral surface of the kidney, a surface that's pointing towards the outside, that is going to be convex, so it bows outward. The medial surface is concave, and there's an indentation in that region in green. That area that's green right there is an indentation that has a lot of tubes going in and out of it. And that's a generic structure that repeats over and over again in many organs of the body. It's known as a hilum. Some people pronounce it as a hilum. Say it however you need to say it to spell it correctly. So we have the renal hilum, which is the indentation on the kidneys that the renal artery, the renal vein, and the ureters are attached to. We also have, in the respiratory system, we had the pulmonary hilum, or the hilum of the lungs. We've also talked about the hilum of lymph nodes in our last unit. This term, hilum, is a generic term that shows up many times over. As we continue looking at a kidney, and I really love this coronal or frontal slice through a kidney because it does such a good job showing the structures. We have renal fascia. Renal fascia is you can think of the renal fascia as the part that holds the kidney in place. We also have perirenal fat capsules. When we have peri, I want you to think around the outside or around the perimeter. What's it going around? It's going around the kidney. The perirenal fat capsule serves a very important purpose in that it physically cushions the kidneys. If you want to punch somebody in the kidney, them in the front, you go from the posterior right here by the floating ribs. And the lower is going to hang down below the floating ribs, and it's a really tender spot on the human body because those kidneys are very spongy, they have lots of tubes in them, and easily damaged. As we look at these renal fat capsules, they also serve a very important purpose because the very best donut recipes call for deep frying your donuts in kidney fat. And I kid you not, it is amazing tasting. My grandma used to make those before she passed away. We also have the fibrous capsule. And when we think of the fibrous capsule, it's going to be immediately wrapped around the kidney. This fibrous capsule, in some extent, can be thought of as a serosa of the kidney, in that it's a membrane, it's a pocket, a bag wrapped around the kidney that helps to separate it from the rest of the body. It protects the kidney from other pathogens that may be in the torso, and it also serves to minimize fluid buildup. So let's say if we have swelling in the abdominal pelvic cavity, that renal capsule helps protect the kidney from that liquid building up in the abdominal pelvic cavity. As we look at the fibrous capsule, it has many collagen fibers that make it up. These collagen fibers are going to merge and become infused with the renal fascia. So the renal fascia binds the peritoneum. The fibrous capsule will then bind to the renal fascia. And this fibrous capsule isn't very tightly bound. There's a little bit of give and take with it, so that when we go from lying down to standing up, our kidneys shift just a little bit. You know, gravity is important. As we age, there's this tendency of gravity to push stuff lower and lower and lower in our abdominal pelvic cavities. As connective tissues break down, the kidneys are no exception. We also are going to have the functional tissue of the kidney. This is a term that I didn't really spend a lot of time emphasizing. Parenchyma is a generic term for the functional tissue of an organ. So the renal parenchyma is the functional tissue of the kidney. This particular tissue is the tissue that's going to make the urine, the secretion that comes from our urines. And as we look at our functional tissues, it forms a C shape, and I'm outlining that C shape right now on the screen. So we have a big green letter C on our screen right now. That's the functional tissue of the kidney, and it's going to be broken up into two big sections. We'll talk about them on the next slide. in the renal medulla. First, let's talk about the center of the C. That center of the C is referred to as the renal sinus. And I'll go ahead and outline that for us right now in blue. Here's our renal sinus, outlined in blue. This renal sinus directly, small indentation turns into a small cavity inside of the kidney. And that small cavity inside of the kidney is going to have an extension and branching of all those tubes that go in. The ureter, and actually let me put a hilum up here. Usually I've taken just a dotted line or imaginary dotted line and drawn it down here and had that orange dotted line represent, I'll say RH for renal hilum. So as we're looking at that renal hilum, that indentation that has all the tubes going in and out, once those tubes get into the renal pelvis, or renal sinus, I should say, they branch off and start forming smaller tubes. For those of you who have had the pleasure of doing a kidney dissection, that perirenal fat pad is going to extend into the renal sinus, and there's going to be a lot of excess adipose tissue in this area to help cushion and separate those tubes within the renal sinus. Let's talk about the parenchyma in more detail. We have the renal medulla and the renal cortex. The cortex, like all the other structures we've talked about that had a medulla and a cortex, the cortex goes around the circumference. So let's go with green for the cortex. It's the outermost circumference of the kidney. And then we'll go with blue for the medulla. And then we have the renal medulla that's deep to the cortex, but superficial to the renal sinus. So as we're looking at these two areas of the kidney, they have different structures and different functions. A good dividing line between them, you can see when doing kidney dissections, are the major blood vessels, particularly the arcuate arteries and veins. This diagram does a good job of emphasizing the histology differences, but in most preserved kidneys, it's kind of a uniform blob or blasé color. It's really hard to visually see the difference in the tissues. We are going to have in our renal cortex and medulla some extensions. Within the medulla, we have these triangles, and these triangles are referred to as renal pyramids. And they aren't necessarily triangles, because remember, we've taken a three-dimensional structure, sliced it to look two-dimensional. In reality, they're more of a cone shape instead of a triangle shape. So that's the renal pyramid, and we typically have 6 to 10 renal pyramids per kidney. And as urine is being formed, those renal pyramids will take the urine and send it to the tip, or the apex of the renal pyramid. And within that apex of the renal pyramid, or the pointy end of the renal pyramid, we'll have a bunch of little openings known as renal papilla that will physically dump the urine into the larger tubes collecting it. We also are going to have some extensions of the tissue that make up the cortex. Those tissue extensions are called renal columns, and I'll go ahead and circle that in blue for us. So the cortex will extend down into the medulla, and as we look at those renal columns, the most notable thing I want to emphasize to you about the renal columns is that they have blood vessels in them. They carry the main or primary blood supply from the renal artery and renal veins out from the renal sinus up to the renal cortex. So when we think of the renal pyramids, they don't have major blood vessels present to them. They have a lot of capillaries, a ton of capillaries, and some arterioles and venules, but those larger blood vessels they don't have. As we're looking at our kidney, there is a lobe, which is one pyramid with this overlying cortex. You can think of it as one pie slice of a kidney. That'll be one lobe of a kidney. I would love it if my touch screen could work here. Does it work? Oh my gosh, it works. It's been like 2 semesters almost, and I haven't figured this out yet. Hold on a second. Let's try that again. This is exciting. All right, hold on. Come on, work. I can draw on the screen. So we have a lobe. That's awesome. So the lobe is the pie slice of the kidney. And then within that pie slice, the kidney includes both the renal pyramid and a renal cortex. And that renal cortex is going to have its renal papilla dumping urine into a small tube collecting the urine. Small meaning minor, so when we look here, we can have a minor calyx, and that minor calyx is this little branching part right here. So we have a minor calyx, one minor calyx per apex of a renal pyramid. And each of those minor calyxes merge together to form a major calyx. So as we are looking at our major calyces, let's go purple for the major calyces. Come on, there we go. We can have multiple major calyces from those minor calyces merging together. And then as major calyces merge together, they form a structure known as the renal pelvis. And we'll go with blue for the renal pelvis. Our renal pelvis is right here. A lot of times I'll be asked, what differentiates the renal pelvis from the ureter? And it's that imaginary dotted line I drew earlier, that renal hilum, that we use to separate the renal pelvis from the ureter. When we look at the histology, from a histology perspective, they're nearly identical to each other. And they both carry the same liquid. The chemical composition of the liquid traveling through them from one to the other is identical. It's all about location, location, location. If we're in the renal sinus, we call it the pelvis. If we've exited the renal sinus, if we've crossed the hilum, we then call it a ureter. So once we get to the ureter, and I'll go with bright red here for the ureter, that's going to be the tube carrying the urine out of the kidney. So let's pause for a moment and have a review question. This is a select all question. Please select the functions of the urinary system. Not all of the functions we talked about earlier are listed. Also, there's distractors out there that other mammals may be true for the urinary system of other mammals. Wait, you can't select all? You can, but hold on. Pause the, all right, let's hold on a second here. This is frustrating for me. Can I pause? I don't think I can pause. I'm going to close it and reopen it. Because I swore I clicked select all. Let's go to the question. Let's go to edit. Apparently it didn't save. There we go. Save. Let's do that again. So hopefully, knock on wood, now you are able to select all the functions. So what are functions of the urinary system, and what are not functions of the urinary system? Make sure you've submitted an answer if you haven't submitted yet. We're down to 5 seconds. All right, so let's look over our results. When we think of functions of a human urinary system, we don't use our urinary system to mark our territories. We do use our urinary systems to regulate blood volumes and blood pressures. On the note of letter A, as somebody who's coached a lot in the locker room, gentlemen, the reason you have locker room. Have you ever been in a girl's locker room and noticed how nice it is, or a woman's bathroom and know how nice it is? You don't have to pee on everything. We do use our urinary system to remove toxins from the blood. We don't detoxify the blood or detoxify the toxins, but we physically remove them from the blood. Our females don't advertise fertility status with our urinary system. Other large mammals do that. We do maintain blood oxygen and ion homeostasis, and no, we don't pee for physical defense. So, our kidneys are pretty small. You know, we have like 2 little bars of soap in our body. And those 2 little bars of soap in our body are disproportionately energy greedy. They use a disproportionate amount of our resources, about 20-ish, 23%-ish. We'll just say about 1/5 of our body's energy goes into these 2 little tiny kidneys. So even though the kidneys are not even 1% of our body's mass, they use up about 1/5 of our body's energy. And the reason our kidneys use up about 1/5 of our body's energy is because there's an incredible amount of active transport taking place. Our kidneys are actively moving chemicals, liquids, molecules against their concentration gradients to take relatively dilute and diffuse blood plasma and turn it into highly concentrated urine. And when I say highly concentrated, I mean relative to the blood plasma. So our kidneys are pretty blood greedy. They use up a lot of our blood supply. And as we're looking at the blood that's entering into the kidneys, we'll start with the renal artery. And this is kind of the classic way to cover it. We start with the blood going in, we talk about the branching structures, and then we end with the blood coming out. So we'll start with the renal artery, end with the renal vein. And let's see if it still works here. Nice. So our renal artery is the entry point of our blood. And as that blood is going into the renal artery, it branches into interlobular arteries. As the name implies, an interlobular artery is in between the lobes. So I'll go ahead and highlight a lobe in blue for us here. Here's one lobe, here's another lobe, and then in between them, we have an interlobular artery that's taking blood through a renal column up to the renal cortex. Once we get to the renal cortex, that blood is going to change directions. And then we'll have a blood vessel that's going to arc around the perimeter of the renal cortex. That blood vessel is known as an arcuate artery. And those arcuate arteries have an arching pattern within the renal column. And then from the arcuate arteries, that arc, we have the cortical radiate arteries. Now, as we look at those terms, cortical radiate, the name tells you where it is and how it's oriented. Cortical radiate arteries, sometimes referred to as arterioles because they're kind of small, are in the renal cortex, hence the term cortical. Cortical radiate arteries are in the cortex. And then how are they oriented? They're radiating outward within the renal cortex. Obviously, they don't extend outside of the kidney, but for the sake of you being able to see, I'm overemphasizing the radiating pattern of the cortical radiate arteries. So we go into the kidney, and then we have an interlobular artery going to an arcuate artery that arches across the cortex. And then we have a cortical radiate artery that's going to extend out to the surface of the cortex, or towards the surface of the cortex. And as that cortical radiate artery is heading towards the surface, it's going to start branching to smaller and smaller blood vessels. We'll have an afferent arteriole, and when we say afferent, that means gaining access to. Just in the last unit, we had afferent lymphatic vessels and efferent lymphatic vessels. The afferent means carrying a fluid towards. Efferent means carrying the fluid or carrying away from. These afferent arterioles go to the nephron. And the nephron is a very exciting part of the kidney. We're going to spend a ton of time talking about the nephron. We have one afferent arteriole, at least one afferent arteriole per nephron. And within the nephron, that afferent arteriole is going to lead to that all-important glomerulus. or a small capillary bed. And this is where most of the action takes place. So the blood eventually goes to a little tiny capillary bed called the glomerulus. From that glomerulus, there are two pathways the blood can take as it's leaving our kidneys. Our blood, depending on what kind of nephron we're looking at, will go to two different secondary capillary beds. The blood will leave the glomerulus through the efferent arteriole that exits through the efferent, and then will go mostly to a structure known as the peritubular capillaries. This is the most common one. So peritubular is by far the most common. But sometimes, in a minority of the situation, instead of that efferent arteriole connecting to peritubular capillaries, that efferent arteriole is going to connect to another capillary bed known as the vasa recta. So the efferent arteriole can go either to peritubular or vasa recta capillary beds. From those capillary beds, we're going to go to cortical radiate veins, to arcuate veins, to interlobular veins, all the way back to a renal vein. So the names in this situation, the kidney, make a lot of sense. If you know the name of the artery, you immediately know the name of the adjacent vein. The tricky part for most students are the capillary beds. We have a glomerulus. Every nephron has a glomerulus. Most nephrons are associated with peritubular capillaries, but some nephrons are associated with vasa recta capillaries. So let's talk about those two capillary beds and these nephrons. I haven't said it yet, and I'm surprised it's taken me this long to say it, so I'll re-emphasize it right now, or emphasize it right now. The nephron is the structural and functional unit of the kidney. So here's the spelling of nephron. The nephron right there, that little guy in the kidney is what physically filters the blood to make the urine. And we have lots of them in our kidneys. Each individual nephron by itself doesn't make that big of a difference. It just produces a couple drops of urine. But because we have so many of them, they all work together, and we end up with a couple liters of urine per day. So as we look at the renal cortex, this is towards the surface of the kidney. So we'll say cortex up here, C for cortex. We'll say M down here for medulla. And then dividing them, we have those arcuate arteries and veins. The cortical radiate, or the cortical nephrons, with their peritubular capillaries, are going to be located in the cortex. So here we have peritubular capillaries. Most of our capillary beds, or most of our nephrons, will have those peritubular capillaries. Every once in a while, though, we'll see a nephron that has a loop extending down into the medulla. And the blood vessels, those capillaries associated with that loop, are called the vasa recta. So we have vasa recta right there, and we have peritubular capillaries over there. So let's talk about the nephron in more detail, that structural and functional unit of the kidney. Our nephrons are numerous. They're legion. We have about 2.4 million nephrons in a healthy adult. We don't need all 2.4 million nephrons. It's not uncommon to donate to somebody or to lose a kidney and have the number of nephrons cut in half. And your blood filtering ability is cut in half. But because our kidneys are so amazing, we can usually get by with just one kidney. I've known individuals that have had only half of a kidney. And these are individuals that had renal cancer. They had to have one kidney completely removed, and then the remaining kidney was cut in half to remove the cancerous tumors. When you're down to half a kidney, or about 500,000 nephrons, then you have to have a highly modified diet to minimize metabolic waste products, because half a kidney has a trouble filtering all the blood in our body, or has a problem filtering all the blood in our bodies. So as we look at our kidneys, we have our nephrons, we have two primary parts. We have the part associated with that first capillary bed, the glomerulus, and that's going to be referred to as the renal corpuscle. And the second part will be associated with the other capillary beds. Collectively, it's referred to as renal tubules. When we look at the renal corpuscle, that renal corpuscle is going to remove fluid from the bloodstream. That fluid will go from blood plasma. Not quite urine yet. It hasn't been concentrated and processed. But it's not quite blood plasma anymore. We're going to have most of the small molecules present in the filtrate, but larger molecules like proteins or bigger things like red blood cells, white blood cells, or erythrocytes or leukocytes, they don't make it into the filtrate. As we look at the renal corpuscle, this first part, we have a couple parts to it. I'm going to actually just skip to the next slide so you can look at the picture while I talk about these parts, because it's a little bit easier to understand if there's a picture to look at while I'm talking about it. So here's a generic renal corpuscle. We have blood going in to our renal corpuscle through the afferent arteriole. So the blood's entering in, and then inside of this pocket or this cavity, we have a bunch of capillaries. Those capillaries are known as the glomerulus. That's that capillary bed inside of the renal corpuscle. And as we're looking at the renal corpuscle, it has a bag on the outside known as the glomerular capsule. For those of you who took anatomy in high school, or anatomy and physiology in high school, I'm willing to bet probably 1/3 of the teachers are still using Bowman's capsule for this term. Bowman's capsule is the old school name for the glomerular capsule. I remembered it really well because my football coach was named Bowman. So we have our glomerular capsule, also known as the Bowman's capsule, that wraps around the outside. So we have that capsule around the outside. And what ends up happening is as blood flows in through the afferent arteriole, it goes to the capillaries of the glomerulus, and it will leave through the efferent arteriole. And that afferent arteriole is much wider in diameter than the efferent arteriole. So we have a pressure gradient buildup. There's a high pressure in the glomerulus because of the differences in diameters. And as this blood in the glomerulus is pressurized, we'll have liquid leaking out of the glomerulus. As that liquid leaks out of the glomerulus, it's going to be processed and will leave through the renal tubules. The end of the glomerular corpuscle, or renal corpuscle, that the blood enters into is the vascular pole, and then the end that the filtrate leaves out of is the urinary pole. It's also worth emphasizing, I'll clear our screen here, that there's a special kind of cell known as a podocyte. Podocyte literally means foot cell. And the podocytes have a lot of extensions coming off of their bodies, wrapping around the capillaries of the glomerulus. And these podocytes can regulate the leakiness of these glomerular capillaries. Can make it so that the glomerular capillaries are more likely to lose fluid when we loosen the restrictions or connections between the podocytes. And I can have an individual little podocyte right here. They're the brown covering over the blood vessels. And if we want to reduce fluid loss, our podocytes can temporarily increase their the number of intercellular junctions that they have, reducing the loss of fluid from the glomerulus. So we've talked about the renal corpuscle. And that renal corpuscle takes blood and removes liquid from it. The liquid becomes filtrate. And now the filtrate is going to be processed in the renal to turn that filtrate into urine. As we look at the renal tubules, we have a proximal tubule, a distal tubule, and a looping tubule for the three primary regions. The proximal convoluted tubule is going to be the one that's closest to the renal corpuscle. Let's think of the term proximal. Proximal means close to the point of attachment. Convoluted used to be a term that most people knew off the top of their heads 50 or 60 years ago. I find that most young people today don't use the word convoluted in everyday speech. It means windy, twisty, turny, indirect. So when we think of a proximal convoluted tubule, it's a windy, twisty tube near the renal corpuscle. Our proximal convoluted tubule in this diagram is right there, circled in green. That windy, twisty tube right next to the renal corpuscle. From the proximal convoluted tubule, we're going to go to the nephron loop. The old school name for our nephron loop is the loop of Henle, and our nephron loop goes down and then back up again. After the loop of Henle, and I'll say loop of Henle because that's, my first two A&P teachers made me learn that term, but nephron loop is the current term. The loop of Henle, or nephron loop, is going to go down towards the medulla and then back up towards the cortex again. And then from there, we're going to jump to the distal convoluted tubule. This is the windy, twisty tube that's farther away from the point of attachment, farther away from the glomerulus. Those are the three primary parts of one specific nephron. One nephron will each have its own proximal convoluted tubule, nephron loop, and distal convoluted tubule. Multiple nephrons, though, are going to share this next structure. This next structure is known as the collecting duct. So we'll have multiple nephrons draining into a single collecting duct. And that collecting duct is going to have a little bit more processing of the filtrate, and then will eventually dump urine out of its opening into a minor calyces, or minor calyx. As we look at the proximal convoluted tubules, the PCT, it's going to come directly from the glomerular capsule, or Bowman's capsule. And we're going to see that in the proximal convoluted tubule, we have most of the reabsorption of important nutrients. Some stuff we don't want to urinate out. We occasionally will have, or will usually have, I should say, amino acids, glucose, and other metabolically really important molecules make it into the filtrate that we don't want to urinate out. We don't want to waste sugar. We don't want to waste amino acids. And in our proximal convoluted tubule, we have most of our reabsorption occurring. To aid with the reabsorption in the proximal convoluted tubule, we need a really high surface area, and we increase that surface area by having a lot of microvilli coming off of the cells of the proximal convoluted tubule. In lab, when you're looking at some microscope slides of the kidneys, it's kind of a hot mess the first time you look at it, because you see a bunch of little white spaces, a bunch of little tubes. You have to train your eye to look for on these microscope slides. Proximal convoluted tubules on microscope slides are going to have these microvilli sloughed off in the preparation process, so you'll see some fibers, some little strands inside of the tube or the opening that's the proximal convoluted tubule. I'm going to switch to green right here because it's not showing up very well up on the board. So right there we can see a bunch of little fibers. Here's a bunch of other little fibers in the opening. Those are proximal convoluted tubules that have those microvilli. As you've probably guessed at this point, DT right here and right here stands for a distal convoluted tubule. And when you look at the distal convoluted tubule lumen, notice how it's not filled with sloughed off microvilli. So there's a visual difference as you're looking at the transverse slices through those two tubes. After the proximal convoluted tubule, the filtrate's going to travel through the nephron loop, aka the loop of Henle, and that's that long descending and ascending portion, that big loop that goes down towards the medulla and then back up towards the cortex. Our loop of Henle, or nephron loop, can be broken up into two segments, two different ways. We can have the ascending, or we can have thick and thin. So let's talk thick and thin first. The thick segment of the nephron loop, as its name implies, is thicker. Physically, it's wider than the thin segment. And structure determines function. The thin segment compared to our thick segment is going to have a simple squamous epithelium compared to simple cuboidal epithelium. And at this point in time, I hope you're at the point where when you hear simple squamous epithelium, your brain immediately goes to diffusion. Thin, super thin layers of cells are really awesome for having diffusion across a membrane. So when we look at the simple squamous epithelium of the thin segments, we have lots of water diffusing out of the thin segment. And the diffusion of water is known as osmosis. We have lots of osmosis occurring in the thick segments of the nephron loop. For those thick segments of the nephron loop, we have the simple cuboidal epithelium. They're really bulky, they're lousy for diffusion, but they're really excellent for active transport. When we get to the thick segments, we're going to have lots of active transport dumping salts into the tissue of the kidney. So if we look at the path of fluid flow, filtrate enters the descending loop. As this really dilute, really watery filtrate is entering the descending loop, it loses lots of water via osmosis. And then, by the time it gets to the bottom of the loop, it's becoming increasingly concentrated. And it's getting to the point on the ascending loop, as the filtrate's heading back up, that it's becoming very difficult to lose water via osmosis. So instead, we shift gears, we shift purposes, and instead of removing water from the filtrate, we're now going to start removing electrolytes from the filtrate to reclaim electrolytes that we need depending on particular chemistry at that moment in time. Oh, I need to talk about a photomicrograph here. I loved this particular histology slide because it shows three clearly different tubes side by side. We have first the thin segment. We'll go red for the thin segment. You can see a simple squamous epithelium around the thin segment. And then we'll shift colors here and go with green for the thick segment. That thick segment has bulkier, thicker cells wrapped around it. That's simple cuboidal epithelium. Visually on the photomicrographs, you can distinguish thin from thin segments two key ways. First, let's look at the loop of Henle. These two tubes are right next to each other. So you need to first look for two lumens that are physically right next to each other on the microscope slide. And then you need one that has a small lumen right next to a big lumen. The small lumen needs to have thick cells, and the big lumen needs to have thin cells around it. So when you see those paired next to each other, that's indicative of a nephron loop on your microscope slide. Let's move on to the distal convoluted tubule. We started with the proximal convoluted tubule. We went to the nephron loop, and then now we're going to end with our distal convoluted tubule, the twisty tube away from the glomerular capsule. Our distal convoluted tubule does not have microvilli. It's without microvilli. So when you look at micrographs of the distal convoluted tubules, you can see a bunch of lumens that aren't filled with sloughed off microvilli. Here with the proximal convoluted tubules, you can see the lumens filled with the sloughed off microvilli. The D for distal has the clear lumen associated with them. Our distal convoluted tubule is also physically shorter. It has physically less turns in it because by this point in time, the filtrate is almost done being processed. It's almost fully become urine. It's the end also of one specific nephron. The DCT, distal convoluted tubule, is the ending point of an individual nephron, and our next tube, the collecting duct, is going to be communally shared by multiple nephrons. So as we look at the collecting duct, we can have multiple distal convoluted tubules all draining into the same collecting duct. And that collecting duct will carry the fluid towards the apex of the renal pyramid. These collecting ducts travel through the renal pyramid, and they all converge at the apex. As they're converging towards the apex, Multiple collecting ducts will merge to form the papillary duct, and then those papillary ducts will eventually drain urine into the minor calyx. When we look at the histology behind this, we're not going to have any more simple squamous epithelium because the fluid here has a relatively high salt concentration. And when I say salt, I don't mean table salt. I mean dissolved ionic compounds. We have a chemical definition here. So we have a high electrolyte concentration. We also are going to have a very low water concentration. And we've invested a lot of cellular energy. We've invested a lot of ATP to concentrate this filtrate that's in the collecting duct. We don't want it leaking out, so we'll have that cuboidal epithelium, a simple cuboidal epithelium, lining our collecting ducts. At this point, once we leave the collecting duct, so we have the filtrate starting in the renal corpuscle or Bowman's capsule. Filtrate travels through all the tubes, gets to the collecting duct. We still call it filtrate as it's heading down the collecting duct, but once we physically get to the end of the collecting duct, we rename the liquid urine. So when we think of the liquid, if we back up a millimeter and we go from just inside the collecting duct to just outside the collecting ducts or papillary ducts, chemically speaking, the liquid is nearly identical. But for the sake of having a clear definition, we'll say once the liquid leaves the collecting and papillary ducts, we call it filtrate. So, or excuse me, we call it urine. Sorry, I misspoke there. So as we're looking at the path of filtrate flow, this filtrate starts at the glomerular capsule and will eventually go to the collecting duct. After the collecting duct, draw a little line right there, it's no longer filtrate. We're now going to rename the liquid urine. And from that collecting duct, we go to the papillary ducts, to the minor calyces, major calyces, ureters, urinary bladders, and urethra. This is worth committing to memory, not only for lab purposes, so you can have a frame of reference, to know the physical path of liquid flow as we filter our blood to having it leave through our urethral orifice. This is a very good thing to commit to memory because there's usually at least 2 lecture exam questions based on this, and there's a lot of lab questions based on this as well. So let's focus on juxtamedullary nephron. As we look at our juxtamedullary nephrons, guys, gals, juxta is a word that's associated with the word juxtaposition or juxtaposing. When you juxtapose something or you have juxtapositioning, you are taking one idea or one concept, either physically or metaphorically, and you're exposing it to 2 different things. These juxtamedullary nephrons start in the renal cortex, and they're also in the renal medulla. So when we think juxtamedullary, they go between the cortex, which is on the top of the screen, C for cortex, and then they go into the medulla, M for medulla. There we go. So they go between the cortex and the medulla. They cross over the arcuate arteries and veins. The key characteristic I want you to associate with juxtamedullary nephrons is a very long, a ginormous nephron loop, or loop of head, excuse me. I'm going to back up a couple slides here. And I want to emphasize here, this is a 2-scale diagram right here in the corner of your screen. We have a cortex on top. We have a medulla on the bottom. Cortical nephrons have a loop of Henle that's pretty dang tiny. So let me change colors here. We'll say cortical nephron in green. There's our loop of Henle. Let's shift colors and go to the juxtamedullary nephrons. We'll do red for the juxtamedullary. The juxtamedullary nephrons start near the medulla, and then their nephron loop goes near to the end of the collecting duct and then goes all the way back up into the cortex. Juxtamedullary nephrons have an incredibly long nephron loop when you look at it being drawn to scale. Around that incredibly long nephron loop, we have a capillary bed known as the vasa recta. And when we look at really long tubes physically, that gives us more time to process the filtrate. And if we can spend more time processing the filtrate, we have the ability to concentrate it even more. So when you look at juxtamedullary nephrons, they have long nephron loops. They make concentrated urine. So when we're in a dehydration situation, or maybe we're in a hypotension, low blood pressure situation, our juxtamedullary nephrons kick in and maximize the amount of water we keep in our bodies and minimize the amount of water that we lose through excretion from the urinary system. We also have those cortical nephrons. Cortical nephrons have really short nephron loops. They do technically cross over into the medulla, just a little tiny bit, but compared to the juxtamedullary nephrons, it's hardly any crossover at all. Cortical nephrons are the most numerous kind of nephron. This is the one that should always be active. When you have very dilute or very whitish or very light yellow urine, that's urine being produced by cortical nephrons. They also are the most common. So most of the time, we have little tiny nephrons making dilute urine. We only use the juxtamedullary nephrons when we absolutely have to, because it's kind of stressful on our bodies. Not only do we use up a lot more chemical energy, a lot more ATP to concentrate the urine with the juxtamedullary nephrons, but if we have very concentrated urine building up in our urinary bladder and in our kidneys, we're much more likely to develop kidney stones from that concentrated urine. Think back to your general chemistry classes. If you have a high concentration of an ionic compound, it's more likely to crystallize out of solution than a low concentration of the ionic compound. So we don't like using those juxtamedullary nephrons because, well, kidney stones stink. Actually, while we're talking about kidney stones, I've had 5 students talk to me about this that have given birth without anesthesia and that have passed a kidney stone. And they assure me, and they all unanimously agree, kidney stones are worse than baby birth. So they're not fun. Let's talk about innervation. When we think of innervation, we think of nerves being sent to the organ to control the organ. The renal plexus is a branch of nerves and ganglia that control innervation to the kidneys. And as we're looking at this renal plexus, it's going to start right here. Let me shift colors here to green. So it's going to originate right here around the abdominal aorta. And eventually, those nerve branches and nerve fibers work their way into the kidneys. We'll talk about this more next week and maybe the week after, about how these nerves can regulate the production of urine. We have some smooth muscle in our kidneys that we can use to constrict or dilate the vessels and influence urine production. So this renal plexus has a sympathetic origin. So it comes from the sympathetic nervous system. It's going to branch off of the abdominal aortic plexus, or all the branching nerves around the abdominal aorta. And the primary purpose of this plexus is to regulate the production of urine flow. Particularly, it's going to reduce the amount of blood flowing into the glomerulus. The less blood we have going into the glomerulus, the less filtrate we'll make. The less filtrate means we'll have less urine. In other words, when you are stressed out, urine production goes down, because when you're stressed out, you have an activated sympathetic nervous system. As we look at this, we also use it to regulate blood pressure. If our blood pressure is low, we reduce urine production to maximize the amount of water we retain in our blood plasma to keep our blood volume up and hopefully keep our blood pressure up. Our kidneys receive some kind of a parasympathetic system or signal. We don't know how it gets there, though. That's one of the big unknowns right now in our human body, among a plethora of other things. How are we doing on time? We have 5 minutes left. Plenty of time for our last review question. Please match the following structures with their functions. And this is kind of a longer question. I'll give you an extra 45 seconds. Please match the structures with their functions. I don't know about you, but I'd be talking to my buddy right now. You're really quiet right now as a class. All right, guys, gals, we have about 20 seconds left. If you have not submitted an answer yet, please submit an answer. It looks like we have a few lingering individuals. It's okay if you get it wrong. This is a participation question. 10 seconds. All right, and we're done. So let's cortical nephrons. Dilute, juxtameasure, make concentrated the fibrous capsule, going to protect our kidney and isolate the kidney. The ureter moves urine away, and finally the renal corpuscle is going to remove filtrate from the bloodstream. Have a great weekend. See you on Tuesday.